Hello everyone. Today we are going to talk about the sexual reproduction in bacteria. In the earlier class we said asexual reproduction or vegetative reproduction. We have seen how the bacteria multiply by binary fission. And here we'll see how bacterial genome can be transferred from one cell to another. How the genomes can be transferred by various means. Though bacteria do not have true sexual reproduction, but bacteria achieve the benefits of sexual reproduction. Bacteria also can get genes or part of genome from other bacteria by various means in nature. So all the processes that will be discussed here, they all occur in the nature and they have been very useful to bacteria to get recombinant type, to generate variation in the genome, to increase their fitness in their ecological niche. It helps them to survive and adapt to changing environment. So genetic changes in bacteria occur in three processes, and the processes are: number one, transformation, and the other two processes are conjugation and transduction. So in transformation, it is simply uptake of DNA fragments or short DNA stretches from outside the cell to inside the cell. So can take a bacterial cell is capable of taking DNA fragments from outside and integrate that fragment into its own genome. So DNA fragments, how they come in the nature, in the surrounding environment. So this can be a question. Now these DNA fragments come from the dead bacteria. The bacteria after death, if the cell is lysed, if the cell is degraded, then the chromosomal material comes out and the chromosome gets degraded by various enzymes, DNase enzymes which are there in the nature. And these DNA fragments are generated and remain in the environment. The bacteria, intact and actively growing bacteria, can take up these DNA fragments and integrate in its own genome. Now, uptake of naked DNA fragments by a bacterial cell is transformation. Now, after the DNA fragment is taken inside the cell, the fragment can either be integrated to the cell, the recipient cell, or it can be degraded. If it is degraded, that means transformation is not successful. This uptake has not been helpful for the bacterium which has received it. But if the DNA fragment is integrated with the chromosome of the recipient cell, then in that case we will say that it is a successfully transformed cell. There are many factors which govern uptake of DNA from the outside environment inside the cell. Transformation depends on the competency of the cell, at what growth stage the cell is, and also various other factors. And because of this, the rate of transformation in nature that occurs is quite low. However, transformation can also be performed in the lab artificially. So the person who first observed or discovered transformation in bacteria is Griffith in the year 1928. This transformation process occurs in nature. It can also be performed artificially in the lab by making competent cells. And transformation is a basic step for knowing the genetic expression of some genes of our interest. The expression of the gene in the bacterium is observed in the lab condition. And very often E. coli cells are transformed, and E. coli cells before transformation are made competent to receive the DNA fragments. So when the DNA fragments and E. coli cells are incubated together in appropriate conditions, then the E. coli cells take up the DNA from the suspension and then get transformed. Now, the frequency of successful transformation can be enhanced in the lab by treating the cells at log phase with calcium and magnesium ions and other things. Now, log phase, it is the exponential growth phase or active growth phase of bacteria. We studied this log phase in the earlier class. So this is about transformation. It simply means uptake of naked DNA from external environment to the bacterial cell, inside the bacterial cell. And there are two fates. Either the DNA fragments can be integrated with the bacterial genome, host genome, or it can be degraded. If it is integrated, then it is a successful transformation. So when this external DNA comes inside, then the bacteria can get some extra trait, additional trait. Okay, then it is a gain for the bacterium. It is a gain for the transformed cell. It has got some extra genes from external environment, and so that this transformed cell will have better survival fitness or better metabolic efficiency. The next process, sexual process, is conjugation. This conjugation process was discovered by Lederberg and Tatum in 1956 in E. coli cells. What happens here? The cells which have got plasmid, F plasmid, they become capable of transferring gene or the plasmid from them to another bacterial cell, which will be the one devoid of F plasmid. Now here we have two living cells involved. One is donor and the other one is recipient. And the donor and the recipient get joined together by sex pilus. We know pilus is a structure, short hair-like structure present all over the bacterial surface, and they take part in conjugation. We learned that in some other classes taken earlier. Now here, when a bacterium becomes donor, for a bacterium to be donor, it must have this plasmid, F plasmid. F stands for fertility. So when F plasmid is present in a bacterial cell, it is called F plus cell. This F plus cell is a donor. It behaves like a male partner in the sexual process, conjugation process. And the recipient does not have F plasmid. So it is F minus cell, F minus cell or recipient, and it behaves like a female partner in the conjugation process. Now this F plus cell, which is a donor, it transmits or transfers a copy of the F plasmid to the recipient or the F minus, and this occurs via a sex pilus. When the cells F plus and F minus cells are close together, then a sex pilus connects the cells together and it forms a bridge through which a copy of F plasmid gets transferred to the recipient. Okay, so both the donor and recipient cells synthesize the complementary strands and restore the double-stranded nature of plasmid, because a single-stranded copy is transferred to the recipient cell. So both the cells initially possess the single-stranded plasmid after transfer. Now this single-stranded DNA synthesizes the complementary strand and it becomes double-stranded and restores the double-stranded nature of the plasmid. Now after transfer, both the cells become F plus. So donor remains a donor, whereas the F minus cell or the recipient, it also becomes a donor. It becomes F plus cell. So this is basically conjugation. Now there are many things which may happen during the process or after the recipient receives the F plasmid. What things will happen? The F plasmid may integrate with the chromosome of the recipient cell. The F plasmid may integrate with the chromosome of the recipient cell. And the frequency of occurrence or frequency of integration of F plasmid with the chromosome of the recipient is 1 per 1 lakh cells in each generation. Now when the F plasmid is integrated to the chromosome of the recipient, then that recipient cell is known as HFR, high frequency recombinants. But this integration process is reversible. The F plasmid can also get removed from the host chromosome. So it is a reversible process. Now when the HFR, high frequency recombinants, are mixed with the excess of F minus cells, then also DNA transfer is initiated because HFR is having the F plasmid. So it will also behave like a donor. Now see here carefully, as we said in conjugation, conjugation happens when F plasmid is present in a cell. That cell becomes a donor. It is F plus which is a donor. Now this cell is F plus. Here we have a plasmid which is shown in green color, and this is the genomic DNA of the bacterial cell. And this is F minus. That means it does not have F plasmid. So it is a recipient cell. If the F plus and F minus cells come together, then sex pillars will be formed. Then a copy of F plasmid will move to F minus cell, and the F minus cell will become F plus. We have seen this much in the earlier slide. Now, when F minus cell receives a copy of F plasmid, then that F plasmid can remain in the cytosol of the bacterium, or it can be integrated to the chromosome of the recipient cell. When the plasmid, F plasmid, gets integrated to the chromosome of the recipient cell, then it is called as HFR, high frequency recombinants. Now HFR, as we said, this integration is reversible. That means the plasmid can also come out of the chromosome of the recipient chromosome. Now this HFR can have two fates. Number one, the plasmid comes out and the host chromosome reverts back to its original composition, and the plasmid is here. So it is simply F plus. So HFR cell, because of reversible integration or because the plasmid gets dissociated from the host chromosome and the plasmid remains in the cytoplasm of the cell and the host chromosome reverts back to its original composition, then it is simply F plus cell, it is a donor cell. But sometimes when the plasmid gets dissociated from the host chromosome, during the process, part of host chromosome can also be attached to the plasmid. See here, in addition to the green colored sequence or stretch, we also have a part of the host chromosome here. Now when this happens, then these cells are called as primary F prime cells. Primary F prime cells are developed from the HFR due to faulty dissociation of the F plasmid from the host chromosome. Now what happens next? So these primary F prime cells, they are also donors. They can also induce conjugation because they have a plasmid with them. Now in this case, when primary F prime cells come in contact with one F minus cell again, F minus cell, then this donor it transfers the plasmid to the recipient. So from primary F prime cells, the plasmid goes to the recipient F minus cell. Then this F minus cell received the plasmid, but it also received a part of the chromosome from the earlier cell. A part of genome from the previous bacterium is also received here. Now this is secondary F prime cell. Secondary F prime cell. So it has the recipient has its own genome, plus it has got F plasmid, and it has also got part of the genome from the previous bacterium. So this part of the genome is also there in its own genome, and here this part is a duplicate part, so it is partially diploid. So this genome has this sequence already because they are all related bacterium. So this genome will be there, this part of the genome will be there, and now it has come from another cell. So for this stretch of DNA, the cell is partially diploid or merozygote. So this cell is secondary F prime cell. Now in this connection we have another term called sexduction. Sexduction or F-duction. That means it is something related to F plasmid. As we have said here just now, in the similar words, in the similar terms, we can say sexduction is something when genes of host chromosome carried by F prime plasmid recombine with the bacterial chromosome. Recombine with the bacterial chromosome. This is called sexduction. Now the third sexual process and the last one is transduction. Transduction process was discovered by Zinder and Lederberg in 1952 in Salmonella bacteria. So this transduction is also a method of gene transfer between bacterial cells, but it occurs through phages, bacteriophages. We all know bacteriophages are the viruses which infect bacteria and kill them. Now, in this process, how the bacterial gene or genome, a part of genome or part of chromosome is transferred from one cell to another. We'll see here. So this is a phage, and typically this phage has head. In this head we have the genome, double-stranded DNA genome, and we have tail. These are the tail fibers. So the phage is specific to bacterium. It has its own host range. Okay, so the phage finds its host. This is the host bacterium, and then it gets attached to the host surface. After attachment, the phage DNA is injected inside the bacterial cell, whereas the remaining part of the phage body remains outside. But in some cases, the phage body can also enter the bacterial cell, but mostly it is the phage genome that enters the bacterial cell. Now after entry of the phage genome, which is a double-stranded DNA, the host chromosome gets degraded. So degradation of host DNA occurs. Meanwhile, we also see that the phage genome is replicated. So after the phage genome is replicated, then progeny phage particles are formed, and when the progeny phage particles are formed in the head, during the assembly process, the proteins which comprise of the phage body and the genome enters the phage head, and in the process mistakenly the phage head can also accommodate some parts of the host DNA, bacterial DNA, some fragments of the bacterial DNA. So here you look that we can see in this phage, in this head we see host DNA, bacterial DNA inside. So this phage head will have its own genomic DNA and also a little bit of host or bacterial DNA here. Now after the release of the progeny phages from the bacterium, the bacteria get lysed actually and the phages are released. Now these phages are ready to attack another bacterial cell. So this is another bacterial cell where there will be another round of infection and the killing or lysis of bacterial cell. Let us see that in the next slide. So this is the phage which is carrying the gene or DNA fragments from the host chromosome, the bacterial chromosome from the previous infection. Now it has come to attack another bacterial cell. So similarly, like in the previous case, there will be attachment of the phage to the bacterial cell, then injection of the DNA, phage DNA inside the bacterial cell. When the phage DNA is injected in the bacterial cell, then we have in that a part of the previous bacterial genomic DNA also. Now this genomic DNA can now get integrated to the chromosome of the cell. So this cell has received a part of a previous bacterial genome, previously infected bacterial genome. Now this cell, bacterial cell has become transduced. It has received the chromosomal DNA from the previous bacterium. So this bacterium has now got a duplicate set of genes for this stretch of DNA which has been transferred by bacteriophages. Now this is a merozygote or partial diploid cell. So this is a transduced cell. Now this genome has come from basically the previously infected bacterial cell. So it is the phage which has achieved its own replication, and in the process it also transferred the part of bacterial genome to the bacterial cell infected in the next cycle. So this is called transduction. So transduction is a means of transferring bacterial genome from one cell to another through bacteriophages. So this is called transduction. Now here when we talk about bacteriophages which are basically the viruses which infect bacteria and kill them, they can go for two types of cycles. Some phages will have lytic cycles. Some phages will show lysogenic cycle. What is lytic cycle? When phages enter the bacterial host, multiply and lyse the host cell, then it is lytic cycle. That means here the phages will enter the bacterial cell, immediately multiply and kill the bacteria. So this is lytic cycle that we have shown in the previous slides. Example of phage like T2 phage. T2 phage shows lytic cycle. Okay, so T2 phage is also known as virulent phage. In another type, lysogenic cycle, here the phage enters the bacterial host and does not multiply immediately, but phage genome gets integrated with the host genome and remain there as a part of the host genome. So this is the stage we call the phage as prophage. Now the prophage remains in the bacterial genome for several generations of the bacterial host, and later the phage genome comes out of the host genome and multiply. After multiplication, the bacterial cell gets lysed and the progeny phages are released, as we have seen in case of lytic cycle. So in the lysogenic cycle, lysis of the bacterial cell occurs, but it is delayed. In lytic cycle, the lysis or the killing of bacteria happens immediately. So in lysogenic cycle, we have seen that killing of bacteria occurs, but it is delayed because the phage remains in the bacterial chromosome as prophage for several generations. One example of phage showing lysogenic cycle is lambda phage. Lambda phage is also known as temperate phage. Now, as far as transduction is concerned, we have two more terms called generalized transduction and specialized transduction. We have already seen that transduction phenomenon was discovered by Zinder and Letterberg in Salmonella in the year 1952. Generalized transduction means any gene can be transferred from bacterial chromosome one cell to another. So whatever examples or diagrams we have shown earlier, that is generalized transduction. So what gene will be taken from the bacteria in the phage head? It is random process. So any gene can be transferred. That is generalized transduction. Whereas in specialized transduction, some specific genes are transferred. As we know that lambda phage is a temperate phage and it undergoes lysogenic cycle, that means the phage after entry in the bacterial host, the phage genome will integrate with the genome of the host and will remain as prophage. But at the time of releasing from the host chromosome, the phage genome will also take a part of host genome, specifically a part of host genome, then it is called as specialized transduction. So in case of lambda phage in E. coli, we have seen that the lambda phage gets integrated to the genome of the E. coli somewhere between gall and bio genes. So when the lambda phage gets released from the E. coli genome, then either gall or bio loci or genes will be taken from the bacterial chromosome. Now when gall or bio gene is taken by the phage genome, then the phage genome will become defective because phage genome will have to miss some of its own DNA in order to get the gene from the host chromosome. So when a gall or bio gene is included in the phage genome during release, a corresponding amount of lambda phage DNA would be missing because phage head cannot accommodate an extra length of DNA in its head. So when a gall or bio loci is included during release, then some of its own phage genome will be missing in its head. So this is called as specialized transduction. There is one phage called P22. It shows both generalized and specialized transduction. So in the Lytic cycle, generalized transduction happens, and in Lysogenic cycle, where the phage genome gets integrated to the host chromosome, specialized transduction happens. Now there is one term called Fuzz Conversion. This is a phenomenon of acquiring new property only because of infection by phage. So when a bacterial cell gets infected by a phage, then the cell acquires new property. That is called phage conversion. For example, Corynebacterium diphtheria, which causes diphtheria disease, it produces toxin only when it is infected by phage. Phage-free bacterial strains are non-pathogenic. So, because of phage infection, this bacterium attains a new property, infectivity, pathogenicity. So, it is phage conversion. This phage conversion is different from specialized transduction. In specialized transduction, phages are defective because in order to accommodate bacterial chromosome while being released, the phage sacrifices a part of its own genome. Now in phage conversion, phages are complete. They just infect the bacterial cell and the bacteria attain new trait. Specialized transduction involves transfer of a gene found in bacterial chromosome through phages. In phage conversion, it involves infection of bacterial cell with the phage only. So that is the difference between specialized transduction and Fuzz conversion. Now that was all about today's lecture. So in this lecture we talked about three important means of gene transfer in bacteria, which are akin to sexual reproduction. Though in real term, in strict sense, these are not sexual processes, transformation, conjugation and transduction, but there are lots of benefits that the bacteria achieve through these processes. So these processes happen in nature, but the frequency of occurrence or rate of transformation, conjugation or transduction is usually low, but this can also be performed in the lab and rate of gene transfer can be enhanced. So thank you very much, and if you have any query about this lecture, you can write to me in the comment box. Thank you again.